Good morning, class. Today we are going to study chapter number 6, that is molecular basis of inheritance. Children, actually factors or genes were first detected and analyzed by Mendel, and subsequently many other scientists by following their pattern of transmission from generation to generation. In 1926, the guess to determine the mechanism for genetic inheritance reached the molecular level. So, the nucleic acids are the first components which were studied. Basically, those macromolecules that present in all living cells. So, nucleic acids are the macromolecules that present in all living cells. Friedrich Miescher was the first person who isolated the nucleic acid from the human pus cell. He called these acids as a nuclein. As it has an acidic nature, so after that scientist Altman called it as a nucleic acid. There are basically two types of nucleic acid that found in the living organism, as we know. That one is the deoxyribonucleic acid and another one is ribonucleic acid, DNA and RNA. Now come to the DNA. What is DNA? We know that DNA is a long polymer of deoxyribonucleotides. The length of a DNA is usually defined as a number of nucleotides or a pair of nucleotides termed as base pair. It contains deoxyribose sugar. What is this deoxyribose sugar? It is a pentose sugar having the molecular formula C5H10O4 with the phosphate group, and the phosphate group have phosphoric acid form the phosphate group and its formula is H3PO4. Actually, this H3PO4 is responsible for acidic nature of DNA. Now, another component is nitrogenous base. This nitrogenous base are the nitrogen containing compound, and mainly it is of two types. One is purines, another is pyrimidines. Purines are the double rings heterocyclic structural compound. It is again of two types present in the DNA. One is the adenine and another is the guanine, A and G. Now come to the what are the pyrimidines. Pyrimidines are the single ring structure compound, and two types of pyrimidines present in DNA are cytosine and thymine. Another thing is, after the composition of these things, we make nucleosides, the compounds formed by the combination of pentose sugar and nitrogenous base. So it is known as the nucleosides. Two things are there: a nitrogenous base attached to the C1 of pentose sugar and an glycosidic linkage. In DNA, nucleosides are formed by the combination of deoxyribose sugar and nitrogenous base. Now, adenine plus deoxyribose sugar is equal to deoxyadenosine. We can say guanine plus deoxyribose sugar, deoxyguanosine. Cytosine plus deoxyribose sugar, deoxycytidine. And last one is the thymine plus deoxyribose sugar, deoxythymidine. So there are four types of these nucleosides. And now come to the nucleotides. The components formed by the combination of a phosphate group with nucleosides When nucleosides attach with the phosphate group, it is called the nucleotides. Means there are the three components are there. The four type of nucleotides present in the DNAs are another deoxyadenosine monophosphate, deoxyguanosine monophosphate, one single phosphate is there, deoxycytidine monophosphate and deoxythymidine monophosphate. These are the four type of nucleotides. Now come to the structure of a polynucleotide strand of DNA. Number of nucleotides links each other by phosphodiester bond and form a long chain of molecules, a very long chain of molecules, that is called the polynucleotide strand. The phosphodiester bond forms between the first and third ends, and each and every strand is known as the one strand is known as the fifth end and another is known as the third strand. Carbon atoms. Actually, it is the position of a pentose sugar. Therefore, polynucleotide strand contains the fifth end and third end. This is all about the today's class of molecular basis. So, molecular basis of inheritance is totally based on the nucleosides and nucleotides. In the next class, we will study about the DNA structure. Thank you. Have a nice day.